Hi everyone, welcome to our PacVec Fall Seminar Series. Today we have two great talks from our trainees, Matthew Ward and Chandler Roe. Today we also have Dr. Neil Vickers from the University of Utah to host the Q&A at the end of each talk. So if you have questions during the talks, please hold them or type them into the chat box. First up, we have Matthew Ward. Matthew is a University Space Research Association postdoc supported by this program and by NASA. He has a keen interest in vector-borne diseases and vector biology and is pursuing a career in global health research, studying vector-borne diseases. Matthew earned his MSPH and PhD from Tulane University School of Public Health in Tropical Medicine. Most recently, Ward was a UJMT NIH Fogarty Global Health Postdoctoral Fellow at UNC Chapel Hill, investigating the genetic variation of T. cruzi, the causative agent of Chagas disease in Peru. So with that, Matthew, you may start sharing your screen.
Thank you. Okay. Okay. Does that look right?
We can see your notes right now.
Oh, that's not helpful. Yeah, let's try that again. How's that?
Okay, looks good now.
Okay, cool. All right. So thank you. Hello, everyone. Thank you for having me. Today, I'll be discussing our ongoing work using mathematical models, inference systems, and mosquito trapping data to forecast West Nile transmission in the Coachella Valley of California. So first off, mosquito-borne diseases are often characterized by complex transmission cycles that are highly variable week in and week out, both temporally and spatially. This makes them uniquely challenging to predict quantitatively at finer scales. So for this project, we wanted to determine if we could use fluctuations in early season environmental conditions to predict higher annual, or to predict the higher annual mosquito infection rates. And we also wanted to determine how scale plays a role in designating areas of risk and whether abatement districts may be able to use our predictions. Justification for this project is that the decision-making process at the abatement district level is often complicated, and resource allocations for public health interventions are often difficult to prioritize. One challenge is that decision makers are often reliant on a limited number of tools and heuristic principles, which may lead to severe and/or systemic errors, but models hold promise for assisting in decision-making for more targeted and cost-effective interventions, improved budgeting, and hopefully optimized surveillance strategies. Briefly about West Nile virus, it's the leading domestically acquired arbovirus in the US with the potential for high morbidity during outbreaks. West Nile has a complex transmission cycle where amplification occurs in birds, but due to the feeding behavior of the vector, we see spillover events into humans, horses, and other mammals. Additionally, West Nile exhibits high annual variability, both spatially and temporally, which further makes decisions pertaining to control efforts more difficult. West Nile was first introduced to the US on Long Island in 1999, and by 2001 had spread across the eastern seaboard. In 2002, 2003, it reached the middle of the country, and we saw the first major outbreaks in the West between 2004 and 2007. This was followed by a recession in total number of recorded cases between 2008 and 11. Then in 2012, we saw the second largest incidence of neuroinvasive cases ever in the US, followed by more variability until 2018, when we saw another high transmission year. But has made it But as is made apparent in this clip, even in low transmission years, there are parts of the country that are still particularly hard hit. Again, demonstrating both the temporal and spatial variability of human cases in the US, and further emphasizing that need for an improved decision-making tools. So our study area for this project is, again, the Coachella Valley of California. Coachella has a historically strong surveillance program with roughly 140 traps per week and roughly 46 weeks per year. It also has a unique environment and geography with its immediate close proximity to the Salton Sea. It has multiple built environments such as golf courses and subdivisions immediately adjacent to agriculture and desert. Additionally, it has temperate winters and largely agriculturally driven hydrology due to its desert climate. The valley has a consistent yearly presence of West Nile with an annual variability of both mosquito infection rate and human case rates in the county. And these factors result in challenges for decision makers, again, making it ideal for a quantitative model to provide structure within the uncertain decision-making process. Additionally, Coachella has a unique climatology that directly affects the valley's mosquito population. For example, in the figure on the left, we have monthly mosquito infection rates by year in black and on the left axis, and then mosquitoes caught per month in red on the right axis. And in the figure on the right, we have historical monthly surface temperature plotted by year, where each line represents a different location across the Coachella Valley. And as you can see, we have a bimodal annual mosquito abundance, which is, you can see here, and a unimodal mosquito infection rate that corresponds to the extreme temperatures in the middle of the mosquito season. This is notable because temperature needs to be warm enough for mosquito populations to develop and for viral amplification to occur. But in the valley, when it gets too hot, the population actually experiences a decline before recovering as temperatures cool. Our model framework consists of three components: a mosquito surveillance system, high resolution environmental data, and a model inference system that uses early season environmental conditions combined with historical annual mosquito infection rates to generate environmentally forced predictions of the annual mosquito infection rates. Our overall development process for the model included creating databases of both mosquito surveillance and fine-scale remote sensing climatology. These originally included data from HER, MODIS, NLDAS, GridMet, and EcoStress. We then used these databases to determine the risk for West Nile virus, and then to see if those early season conditions could be used to predict that annual mosquito infection rate. Finally, we hope to eventually produce a decision app for West Nile virus risk that could be used at the abatement district level. To assemble our databases, we downloaded and processed and assembled all those environmental remote sensing data sets. We matched and processed 15 years of mosquito and West Nile data from the Coachella Valley to the environmental data. And then we evaluated the different spatial scales of each data set and evaluated correlation between environmental variables. We ended up choosing evapotranspiration and surface temperature for our initial modeling efforts. And then we worked to understand the influence of the environmental indicators on the population dynamics portion of the model and how variability at the different spatial scales might affect the model. Our final environmental database consisted of NLDAS data, which has a 13 by 13 kilometer resolution at an hourly frequency, at GridMet, which has a 4 by 4 kilometer resolution at a daily frequency, and EcoStress, which we still plan to use, which has a 70 by 70 meter resolution at roughly a 4-day overpass frequency. And these together give us a highly resolved combination of land surface temperature and evaporative stress index through evapotranspiration. A little bit about EcoStress, which is sort of the highlight of this high resolution, and it's got the finest spatial scale, is that EcoStress is a thermal infrared radiometer aboard the International Space Station. that accurately measures the temperature of plants at the finest scale ever acquired from space, that 70 by 70 meter grid. And but with this fine scale comes questions around how to interpret both the environmental and surveillance data, both temporally and spatially. For example, these plots depict historical annual average infection rates per 1,000 mosquito tested between 2006 and 2019 at the yearly, monthly, and then weekly timescales matched and projected at the NLDAS grid level. So these figures demonstrate that there is a high variability both spatially between the grids and temporally as we descend from the annual to the monthly to the weekly time step. This leads us to the question of what scale are most appropriate to capture the true state of transmission. Similarly, we observe significant variability in infection rate at the different spatial scales of the environmental data. For example, on the left, we have the historic annual infection rate per NLDS grid cell. And on the right, we have the same annual infection rate per grid met. And you can see the variation of the grid met cells within the corresponding NLDS cells for the same year. To partially address this and through inflation associated with mosquito infection rate data, we set thresholds around the temporal periods of risk. In the figures on the left, we have the historical frequency of positive traps in blue with the historical frequency of negative traps in red. And then on the figure on the right, we have the cumulative percentage of positive pools in the red line ascending here, and historical average climatology or temperature in the solid black line here. And that a threshold set at 15 degrees Celsius, and then another threshold here for 5% cumulative positive pools in this dashed red line. From these data, we observed that it is actually a very short period of time that defines risk in the valley. So we set temporal thresholds for data simulation at the 5% cumulative positive mosquito pool threshold and an average temperature of 15 degrees Celsius. These thresholds resulted in us using January through August environmental data to predict the annual mosquito infection rate calculated at each grid scale between May and October of the same year. For our model development, we evaluated multiple models and settled on a multi-model inference system, including 4 environmental variables and the temporal thresholds I just mentioned. We used the MLE, or the annual MLE per 1,000 mosquitoes calculated at each spatial scale as our outcome, and the GLMM ADMB package in R. We ran the package with a hierarchical model with a random intercept for each spatial scale grid cell and a negative binomial with 0 inflation as the link function. We then weighted and ranked the models, selecting the top models whose AIC weights sum to 0.95 or greater. So from all the combinations we ran, we really wanted to see if anything stood out. Was there a consistent pattern at our different spatial scales that suggested different combinations were driving transmission in the valley? So in all, combinations yielded 9 significant models at the NLDAS scale, with one model consistently scoring significantly better than the others when ranked against each other. The coefficients and relative importance of each variable in the model shown above indicated that a drier January, followed by a warmer than usual February, March, and most importantly, April, best predicted the annual mosquito infection rate. As we descend or move down in spatial scale to the grid met level, the combinations yielded 21 significant models, with yet again one model scoring significantly better than the others. This model indicated that a drier February followed by a warmer April and June, with a cooler July as the seasonal drivers of transmission. And furthermore, this model demonstrated that even at the finer scale, a similar general climatology or temporal climatology held true as with at the NLDAS level. So when we look at this from the biology standpoint, behind these results, we build sort of a narrative that suggests that transmission builds over time, increasing the overall mosquito infection rate as West Nile spreads north through the valley, and that an optimal transmission season would look something like a drier winter where birds migrating into the valley arrive in and are sequestered to fewer sources of water, primarily in the south around the Salton Sea. The same area is where the mosquitoes would be at the time of year. And in such close proximity, mosquitoes are then feeding on the birds. And if it was not already present in the system, West Nile is introduced. Then the valley begins to warm earlier than usual. Mosquitoes' development speed up, population builds faster. There's a shortened EIP of the virus, and transmission begins to spread out northward in the valley. This is followed by a cooler than usual summer, where the mosquito population persists longer, avoiding the high temperature associated declines seen in other years. And transmission can then continue, again, increasing the overall annual mosquito infection rate. Our next step in the project is to validate our model using a leave 1 year out strategy, followed by using data from, or integrating data from EcoStress to see if we can't further go down in scale. But at that scale, there are challenges with the sensitivity of trap data, as well as at that fine scale, microclimates will likely play a significant role, sort of where small wet areas within dry areas become much more important. And an example of this fine scale EcoStress data is in the figure below, where we have mean evaporative transpiration during early season on the left and late season on the right, with the red X's indicating trap locations. And from this, we can see a marked difference in the hydrology between these two periods, with the darker the shade of blue-green indicating evapotranspiration and more moisture in the system. Additionally, we would like to continue to refine and tune our model, comparing different indicator combinations. And then finally, due to that unique climatology in the valley, specifically the temperate winter, we would like to investigate the effect of the end of the previous season on the current season. So does that second abundance peak that we see in the data represent population recovery after the high temperature events of the summer? And do those recovery periods have any effect on the following season's transmission rates and infection rates with West Nile? And thank you. I'd like to express my thanks to our funders, especially PacVec and NASA and the EcoStress project for specifically supporting my work on this project. And thank you guys for your attention.
Thank you, Matt. That was a nice presentation. If there are any questions, feel free to either raise your hand or unmute and just speak up, or type your questions into the chat and I will read them for Matt. So let me see. Matt, if you maybe you could end your presentation so that I can see all the speakers.
Sure.
Thank you. Let's see. Anybody got any questions? Okay, I have a couple for you, Matt. So I'm not a modeler, but this is pretty interesting work that you've done. Presumably your models, because they're based on the data, accurately predict which years have been big years for the West Nile virus in the Coachella Valley?
So far they do, and that's why we're going to further validate with the leave 1 year out, because the real challenge is it's easy to predict, or it's not easy, but we can predict the null model, which is just the average mean, and it's trying to figure out which drivers which are the drivers for those different than average years.
So can you explain the leave 1 year out approach? That's not something that I'm not a modeler, like I said. So how does that work to validate the model?
So we would use the we would use all the data except for without the year in question that we're trying to predict, and then we'd run the prediction and see how well it predicted the actual annual MLE that we left out, and try to do that for all the years and see how well it scores.
Okay, cool. There's a question in the chat. Let me tell you what that is. So Chris Barker has asked a question. He says, great presentation, Matthew. I have wondered whether the midsummer drop in Culex abundance is attributable to reduced adult survival during the very hot months, or is it reduction in larval habitats? Have you used any remotely sensed data to determine how the mosquito dynamics relate to the larval habitat availability, primarily irrigation of agriculture and later flooding of duck clubs?
No, so that's something that we want to do going forward, because as I mentioned, Coachella has got that sort of built infrastructure with, for instance, there's a string of golf courses that run up the middle of the valley, and then mixed with those agricultural areas, which get different, which drives the hydrology based on irrigation. And then early in the season with the flooding of, as you mentioned, duck clubs, because basically earliest season is duck season, it's migratory waterfowl season. And so, yeah, that's something that we want to look into.
Okay, thank you. Next question on the chat is from Jennifer Henke from the Coachella Valley MVCD. So these are the real boots on the ground here, Matt. So
she gave us the data.
Somebody who knows exactly where the weaknesses are. Okay, great presentation, Matthew. Given that our environment might be what others could experience with continued climate change, what advice do you have for other districts in more temperate climates?
My advice would be prepare now, I guess, as things do continue to dry out, where we see drier summers and less, as Chris mentioned, less larval habitat, less more concentration of birds and mosquitoes. We're likely to see more spread of West Nile, and so I guess take that into mind and build your abatement strategies as such, or look at Coachella Valley as an example.
Yeah, it's a good point, because I'm up in Salt Lake, and I was kind of struck a little bit by how the Coachella Valley is a sort of a similar kind of ecosystem to to this Great Salt Lake area, and we have freshwater flooding for duck clubs and so on and so forth. Mm-hmm. So there's another question in the chat which I'll get to in just a second, but have you thought about looking at how your models might apply to other areas, like that are comparable in terms of topography and hydrology, like the Salt Lake area? Because we had a big like you probably experienced this year, it was a pretty big year for West Nile virus.
Yeah, no, so that's something we definitely want to do, and I think we need to validate first using that leave 1 year out strategy, and then look towards seeing if we can apply it to another ecosystem, whether it be similar to yours or similar to the Coachella Valley, or as Karen mentioned in the next question, ecosystem that's different, and see how well it would do, since Coachella is so unique. And then to answer Karen's, so wetter and drier, we did it over the course of the 15 years that we had data. And when I say wetter and drier, you got to bear in mind that we're in a desert, so it's all relative. It's relative to the climatology of the desert, not per se Coachella versus wetter, say, than like New Orleans or something. It's within relative to the climatology there.
Okay, any other questions for Matthew? All right, well, thanks, Matthew. That was a great talk. And Celia, shall we move on to the next presenter?
Yes.
Thank you, I appreciate it.
So next up, we have Chandler Roe. Chandler is a PhD student in informatics and computing with an emphasis on bioinformatics at Northern Arizona University. Chandler's work in Dr. Jason Saul's lab has focused on population genomics of infectious diseases, with a particular interest regarding the genomics of the vector-borne disease Onchocerca lupi in Northern Arizona. She hopes her research conducting targeted and controlled sampling of hosts and vectors of O. lupi will directly inform vector control units to employ black fly control efforts near high-risk areas, thus breaking the transmission cycle. Once completing her PhD, Chandler plans to establish her career as a researcher in parasite genomics in academia. Chandler, you can take it from here.
Okay, can you see that, Celia?
Yes.
Okay, great. Yeah. Hi, thank you for having me. My name is Chandler Roe, and I am a PhD candidate in the School of Informatics and Computing at Northern Arizona University, and I'm under Dr. Jason Saul and Dr. Crystal Hepp. And today I just want to really briefly walk you through our ongoing project where we're focusing on host and vector screening for Onchocerca lupi. If you haven't heard of Onchocerca lupi, I'm sure you're not alone. This is a newly emerging filarial nematode, and it causes a disease called onchocercosis. It's a vector-borne disease, and it's found in southwestern United States, as well as regions in Europe and the Middle East. And the putative vector for this parasite is thought to be a black fly species. This is largely based on vector knowledge of other closely related Onchocerca species. So a paper in 2015 identified Simulium tribulatum as a putative vector for Onchocerca lupi, but active transmission has yet to be reported from any vector species. So this parasite has been reported in dogs, cats, coyotes, wolves, and humans. And to date, I think there's been 12, I wanna say there's even a new case recently of a human case of Onchocerca lupi. So 13, 7 of them have been in the southwestern United States, and 4 of those have been in Arizona. So this disease is also non-reportable by vets. It makes defining the geographic distribution pretty difficult. So there's no current commercial diagnostic. We have extremely limited treatment options, and there's no cure for the parasite. Earlier this year, we published a paper in the Emerging Infectious Disease Journal where we identified coyotes as a reservoir for Onchocerca lupi. And this map is a map of Arizona, and we're just looking at the darkest colored counties. Those are Navajo and Apache counties, and those are the highest positivity rates for this study. What's interesting is that these 2 counties overlap with the human cases in Arizona and a high number of canine cases. So based on these data, we decided to focus our surveillance efforts in these specific counties. I want to really briefly walk you through kind of what people currently think is the life cycle of Onchocerca lupi, but it's unknown still. So this is taken from Onchocerca volvulus, which is the human version of this. So yeah, take it with a grain of salt. But basically, Onchocerca is a vector-borne disease, and it has these 2 stages in its life cycle, which is the black fly and the host. And so first, black fly takes a blood meal from an infected host, and it ingests microfilaria or larvae. Next, the microfilaria penetrate the midgut of the black fly and migrate to the thoracic muscles, and they mature there. And at this stage, they are considered L1 larvae. The L1 larvae then travel back to the head and proboscis of the black fly, and here they become L3 larvae and infectious larvae. So at this point, the black fly takes another blood meal, and the L3 larvae enter the bite wound, and then we move into the host stage of the life cycle. So in this case, instead of the human stage, it can be the human stage, most likely the canine stage. The L3 larvae mature into adults and form subcutaneous nodules. And Onchocerca was named after onco, as in tumor. So these nodules look like actual just tumors sitting up on the eye. And then at this point, the adults produce this unsheathed microfilaria that are found in the skin and lymphatics of connective tissue. And so a single adult female without a male presence can produce 700 microfilaria a day. So you can imagine the discomfort this disease causes these animals. So our main goal for this project was really to increase the host and vector surveillance so that we could identify the actual O. lupi vector. We wanted to understand the parasite distribution and prevalence, as well as identify regional hotspots of the disease. And so this will in turn allow us to have these informed and targeted vector trapping sites, which would increase our chances of identifying vector species and demonstrate active transmission. And we also want to perform blood meal analysis in order to identify other potential host reservoirs that are currently unknown we're just not looking at. So the Navajo Nation spans both Navajo and Apache counties, and it's comprised of over 27,000 square miles, and it extends into Utah, Arizona, and New Mexico. So we started this project in 2019 through a collaboration with the Navajo Nation Veterinary Management Program. So the NNVP estimates they perform about 600 surgeries annually. So for this project, the NNVP collected skin samples and stored them in ethanol, and they also collected metadata on all the animals if possible, but a lot of these were strays. Samples were then transferred to NAU, and we performed a DNA extraction and screening for O. lupi using a previously published cytochrome oxidase 1 mitochondrial gene PCR assay. So these primers are not specific to Onchocerca lupi, so if we see any bands on a gel, we sequence the amplicon to confirm that it is Onchocerca. This part of the project is providing us the data to conduct the targeted vector trapping. For this study, we also created a public website for the community members to learn about the project, but also provide information on the parasite. And then on the back end of the website, we created this private online database for community stakeholders, veterinarians, researchers, so that they could track sample metadata and results. This database also populates a map with positive sample locations, so we can easily visualize any O. lupi positive clusters if we find them for our next phase of our project. So using these high-risk locations, we then set CO2 baited mosquito traps for vector surveillance. And then the trapping season for this is really May to October, but we set traps weekly from May through the end of August. And then the plan was to morphologically sort black flies and then sex them. Not that we expected to see many males using a CO2 baited trap, but just in case. And then the females would be screened for Onchocerca lupi using that same CO1 assay as the dog samples. And then again, we'll sequence to confirm that this is Onchocerca lupi. So our collaborators, the Navajo Nation Veterinary Management Program is run by Dr. Kelly Upshaw-Bia. And it consists of 3 veterinary clinics and 1 mobile unit that serves the Navajo Nation. So over a 2-year period, they collected 407 skin samples from the interocular frontal area of the animal's head, which you can see highlighted in red in the photo. So these samples are randomly collected from spay and neuter clinics, as well as any animal undergoing a surgical procedure with owner consent. So these samples were stored in 80% ethanol and then shipped to NAU for processing. And once we received them at NAU, we digested the skin samples overnight, and the DNA was extracted using just a QIAGEN DNAZ blood and tissue extraction kit. Samples were then screened using the CO1 primers, and the resulting amplicons were sequenced to confirm. We identified 41 positive samples of O. lupi, which is, and I do want to say that all of these dogs, they had 0 symptoms for this disease at this point. So it's really just random collection with no symptoms. And so we see about a 10.1% prevalence so far, which is a lot higher than I expected, honestly. And using this data, we were then able to identify multiple Onchocerca lupi positive clusters. And so for privacy purposes, I can't include any location data at the request of the Navajo Nation, but I can say that we identified positive clusters within really diverse environments, such as farmlands, creeks, washes, and maybe most interestingly, near a dry wash bed. So these locations are directly informing the second phase of our project, vector surveillance. I feel like I've said that 3 times. Okay, so also just something we got to do because of the PacVec, it was really exciting for me, was starting to look at the mitochondrial genome for Onchocerca lupi. So the samples we get from animals are extremely complex DNA samples, and the majority of the DNA belongs to the host and not the parasite. And so with the dog samples, all we can really do is say that they are positive or they are negative, but we can't really infer anything else. So we were able to sequence and assemble a complete Onchocerca lupi mitochondrial genome, which we published. And this allowed us to then selectively amplify and sequence the mitogenomes from 22 of our O. lupi positive complex skin samples so that we could infer population structure. So this sample set did span 11 years. And what we found was a single mutation out of 13,766 bases of the mitochondrial genome. So I really did not expect that low of diversity, which is just so surprising. So this basically, that's what this picture is over here. You're seeing, hopefully you can see my mouse, but we have a read pileup, and the reference is a dog from 2010. It's a T here, and this sample has a C. So from this, we can really tentatively say that we think that there was a single recent introduction of Onchocerca lupi into the United States, followed by a rapid radiation. However, we really need to do whole genome sequencing to corroborate this hypothesis. But I was still extremely excited getting to work on this. Okay, so we're using a very targeted and informed vector trapping approach, and that's in hopes of identifying the vector species as well as active transmission. So to do this, we have, let's see, we obtained the help from Dr. Kelly Upshaw-Baya and Olivia Holliday. Olivia was amazing. She took me all around the reservation for the entire summer. And the two of them got permission for vector trapping from local ranchers, farmers, residents, and government agencies as well. So within these high-risk locations that our study had already identified. So we started phase 2 in May of 2021, and we set CO2 baited mosquito traps that ran for 7 days in 15 different locations. And certain locations we set traps at for multiple weeks, depending on how hot we considered a location. Or in the case of a couple of weeks, we had our CO2 tanks tip over, and so they weren't emitting any CO2. So we resampled in those locations multiple times. But this picture shows our trapping setup. We were in a gulch where cattle were roaming, so we had to put the trap in a way where it wouldn't get trampled. This trap, the setup is incredibly large and really heavy, and I did not think about that going into this at all. I was just thinking a solar panel would be good and we wouldn't have to have a battery for it, but we do. And the battery had to be 50 pounds. And then our CO2 tanks were also 50 pounds. And so hiking these around on the res all summer, it was an incredible experience, but it was extremely tiring. And I'm really thankful that Olivia was willing to do that with me. But in hindsight, I'd probably change the design a little bit. Let's see. So we had one site that I termed the hot zone, and it's where we had 3 positive dogs within a 2-mile radius, and there were multiple water sources nearby, including a farm line, a community water reservoirs, a lake, and then a currently dry, but it was sometimes wet wash. And so we trapped at this site, our hot zone site, for a couple of weeks, but then we actually followed the wash down about 35 miles south to a farm. Got permission from the farm owner, and we trapped there as well, even though there were no positive dogs nearby. And so this was our only non-targeted trap site out of the 15. And I just want to show you the landscape diversity of our 15 different trap sites. So it spanned from 100+ heat in the desert with no water to dry wash beds to cool pine forests to cattle ranches. They all had different types of water sources, such as cattle troughs, canals, washes, creeks, lakes, but none of the informed sites had moving water that would be necessary for black flies. And so at the time it was a little alarming, but I thought we'd still probably be okay. So over the 4 months, we trapped at 15 different sites and we collected over 5,000 insects. And upon sorting all of them, we found exactly 0 black flies, which was pretty panic-inducing at the time. So at this point, we decided we really needed to change our strategy. So we started separating insects morphologically and screened 1,622 insects for Onchocerca lupi. And we identified 57 positive samples from 12 of the 15 sites we trapped at, including the non-informed trapping site where we followed the wash down south for 35 miles, which is really crazy. And in fact, at that site, we caught over 2,000 biting midges. So of the 57 positive samples, 20 of the positive samples were from biting midges, and these were in pools of 50 because of just the sheer number of midges, and they're just so small. Started pooling them in groups of 50. Most of the biting midges are either, let's see if I can say this, Ceratopo no, I can't do it. Ceratopomene species, I'm sorry, I'm butchering it, or Culicoides species. So Culicoides species have been shown to transmit a cattle version of Onchocerca in Africa. So that was really exciting to me. And then we also identified 19 samples as Stomoxys calcitrans. So this species has also been implicated as the vector for cattle, for the cattle version of Onchocerca in Africa. And 14 of those 19 samples were in the heads of the flies. So that suggests active transmission. We were able to, any of the flies, we process the head separate from the body in hopes of seeing active transmission, except with the midges because they were just so small and we just had so many of them. So this is the first time anyone's shown active transmission for Onchocerca lupi, but it's also the first time this Onchocerca lupi has been identified in this vector. We had 13 samples that I was not able to identify as of yet. So this was using a 16S target, fly target. So I'm going to, well, hopefully I'm going to use another molecular marker that will actually be able to identify those 13 samples. And then we also had one Musca domestica that was positive, and that was in the head as well. And I read that those can be opportunistic blood drinkers, but I'm not an expert at all, so I definitely want to talk to a few people about that. And then I had 4 positive flies that are most closely related, based on 16S, are most closely related to a flesh fly or a blowfly. And that was at 96% identity. And so I would by no means say that these are flesh flies or blowflies, given that nucleotide identity. But it is just the closest relative so far that we've identified. And what's interesting is one of those flies also had O. lupi in just the head of the insect as well. So finally, we also identified an unknown Onchocerca species. So we sequenced the CO1 gene, and that shows that it was unique compared to what is in the database. And it most closely hit to Onchocerca gutturosa, which is a cattle version of Onchocerca, at 93% nucleotide identity. So hopefully we can wrap this up in the next 2 months, but we have a few things we're still looking into. So first, I really want to discern the other Onchocerca species. It's been suggested that maybe this is the horse version of Onchocerca, Onchocerca cervicalis, but it is not represented in the database using the CO1 gene at all. But there is a gene representative of the 5S. So we're going to target that, sequence it, and see if it looks like it is cervicalis or if it is a novel species of Onchocerca. We also really want to demonstrate active transmission with our biting midges. So we still have a couple thousand biting midges left from one of the positive sites, which you can see in this tube here. I think we have probably like 800 midges in that one tube. And so now what we found is, so we have to actually go in and cut the heads off the midges. So we're doing that under the microscope. We're still pooling them in sets of 50. And then extracting the heads separately from the bodies. But what we noticed was that the beads that we're using to break open the insects are not hitting the heads. The heads are just too small. So we adjusted that by using liquid nitrogen and an actual mortar and pestle. And then you can see that in the third picture. We're actually able to finally grind up the heads more. So we're hoping we will be able to see active transmission within the biting midges. And then we still need to identify the remaining unknown fly species. And so now I'm going to use a CO1 fly target instead of the 16S and see if we can get any closer. And then finally, I want to do the blood meal analysis. And so it's kind of interesting. There's a moratorium on sequencing Native American DNA. And so we can't take all of the samples forward unless we know there is no human DNA in them, which there could be if they're biting humans. So we're going to screen all of our samples first using a human-specific ALU target, and it's going to just be an old school PCR and run a gel. And if we have a band, then we know human is in that, and we won't mess with that sample for blood meal analysis. Any that don't have the human signal, then we will go ahead and process for blood meal analysis using published mammalian primers. And I think that's pretty much it. If anybody has any questions.
Hi, Chandler. That was a really interesting talk. Thank you. There are a couple of questions in the chat, and so I'll read those out for everybody. Chandler, super interesting talk. This is from Chris Barker. If O. lupi was recently introduced into the US, where would you speculate that it came from, and how could it get here? And he says he's sorry if he missed this, but he's thinking about the potential history of coevolution of vertebrate host and parasite. And this is another question: is the transmission the result of infections in wild or domestic canids in other locations where O. lupi occurs? So there's two questions here. The one is, where would you speculate it came from, and how could it get here?
I don't know if I can say like where it came from. So we know that it's in Europe, Eastern Europe. We know it's in some of the Middle East, but it makes sense that this would be movement of dogs, of owners from some of these places into these new locations. I think that makes the most sense versus anything to do with the wildlife necessarily. But whether it came here from there or from here it went over to there, we won't know yet until we do some more whole genome sequencing.
And then the second question is regards to sort of the coevolution of vertebrate host and parasite. Is transmission the result of infections in wild or domestic canids and other locations where O. lupi occurs?
So there has been the first report of Onchocerca lupi was in a wolf in 1967 in the Republic of Georgia. And to my knowledge, that is the only other wild report other than our coyote study.
Okay, thank you. And then next question is from Derek Lake. He says, great presentation, Ms. Roe. Is there any possible connection to the Colorado River as far as a means of transporting vectors containing O. lupi?
I think absolutely. Yep. So we definitely have some other areas that I'd like to really target, but I don't know. I definitely think the Colorado River is playing an important part. We see that we have dog cases up in Colorado, and if you follow the river down, you can see cases all along it. I think that just given the position or the climate, that's going to determine what the vector is going to be. But along the Colorado makes a lot of sense for the black flies.
Okay, I have a couple of questions. So one relates to just the black flies in general. So you set out a bunch of traps and you didn't capture any black fly. Are there actually any there? Do you have they been reported from there before, and are they known to be in that sort of habitat?
I think in certain areas it probably makes sense. So there's the San Juan River runs right through, and I think probably in those areas I think we probably would have seen black flies there. There's no reported anything as far as that region. For what insects are out there, but I think we couldn't get any permission to get into right along the river in one location. So
right,
maybe next year.
Yeah, that's an interesting area. Do you when you just a practical question? So yeah, I know how heavy those deep cycle batteries are because I've used them in the field too. I don't know how far you had to carry that, but they are heavy. Mm-hmm. And so one of my questions was how long did you leave the traps out? I don't know whether you mentioned this or not. What was the trapping period? Were you doing it for a week or?
It was for a week. Yeah, that was the soonest. I think in one day I would end up driving upwards of 700 miles, and so we really could only do that once a week. So we had to make sure it would run that long.
And it's just it looked like a sort of a BG sort of sentinel trap. Yes. with where you pumping the carbon dioxide in.
Yep. Yeah, okay. Exactly.
Yeah. So of course you had to hike the tank in as well. Hopefully you didn't have to hoof those things too far.
A couple of the sites we did, but I learned from that and brought appropriate gear next time. So.
Right. Okay. Make sure that you can take a cart with some wheels on it or something into your field sites. That's what you need to do.
Seriously.
I totally sympathize though, having been out in the field myself. In the desert. So yeah, that's good stuff. Are there any other one other question. So when you're looking at the insects that you do catch, are you taking those down to species level or where you can, or are some of them sort of unknown? Because I think most of them you had were at the family level, right?
Yeah, I had two. I think I had the Chilocoides, that's the genus.
Yeah.
So had that, but okay, what we were doing is I'm not equipped. I'm not an entomologist. So what we did was we took pictures of every single insect that we extracted from, so we could go to an entomologist with questions, and then molecularly, I was really hoping molecularly I would be able to get those down further, but so far I haven't, which I was really surprised at, but I was just very naive, I think.
Thank you. Okay, there's one more question's come up in the chat here from Coachella Sherpa. Hope I said that correctly. Were the collection sites mostly open, as in the picture, or were some in wooded areas too?
Somewhere in wooded areas, yes, absolutely.
Okay, so, but it was quite varied habitats though, right? I mean, that's what you were looking at.
Extremely varied. Just wherever we found a dog that was positive, we could get near that site. That's where we wanted to trap. But we also had the issue with gear being stolen, leaving it out. And so we wanted it always to be on somebody's land that we knew and that they knew that it was there.
Right.
So it made it a little more difficult.
Yeah, those are another hazard of field work like that. Things go missing or well, when we did field work in California, we always noticed that things used to get shot at by people out in the so another thing that you have to be careful about. But any other questions for Chandler? Okay, I'm not seeing any. So Celia, do you want to wrap up?
Sure. Thank you, Matthew and Chandler, today for your presentations, and for Dr. Neil Vickers for hosting the Q&As. This wraps up our 4th seminar of the series, so please join us for the next one on November 23rd, same time from 12 to 1 Pacific time. Thank you and have a good rest of your day. Bye-bye.